Um, Thank you for attending this webinar on multiphysics modeling of microfluidic systems. This is part of Verist Engineering's free webinar series where we discuss our capabilities and the types of projects we work on with our clients. So my name is Matt Hancock. I'm a partner here at Verist Engineering. I received my PhD from MIT, and I consult in fluid mechanics related to product design and performance with core areas including microfluidics, surface tension and wetting, heat transfer, species transport, and mixing. Prior to joining Verist, I was a visiting scientist at the Broad Institute of MIT and Harvard. Before that, I was a postdoc at Penn State for a very short time in a biointerface lab. Before that, I was a postdoc at Brigham and Women's Hospital and Harvard Medical School. And before that, I was an instructor in the Department of Applied Math at MIT. Verist Engineering is an engineering consulting firm headquartered in Boston. We provide specialized subject matter expertise across engineering, material science, and physics to help our clients efficiently solve their most challenging problems, accelerate product development and manufacturing, and deliver better products to market. We bring academic level expertise on an entire industrial timeframe, drawing on a wide range of capabilities, including fluid mechanics, multiphysics simulation, heat transfer, electrochemistry and reacting flow, solid mechanics, polymer and materials testing, and materials engineering. We use a range of tools, both computational and experimental, to perform analyses and develop guidance and recommendations for our clients. So in today's talk, we'll highlight microfluidic examples, including those from our industrial partnerships, to demonstrate how we accelerate and enhance product development. We take a hierarchical approach to problem solving, employing an array of computational and experimental tools with varying levels of sophistication. Specific to our modeling strategy, we start by laying the groundwork with hand drawings, back of the envelope calculations, and these help us identify key physical mechanisms at play and pinpoint the key parameters and parts of the system that are critical to its performance. We then proceed with model development and ensure that the type and scope of the model can address the important outcomes desired by our clients. We also decide how the model will be verified and validated. So once the model has been developed, verified, and validated, we compile model results into clear, actionable design guidance, and we work with our clients to implement our recommendations and develop plans for additional iterations, optimization, or analysis on other parts of the system. We always keep the outcome in mind. What is our client trying to develop? What are their roadblocks? And how can the model or models address the key questions to reduce product development time? So now we've reviewed our modeling methodology, I'd like to highlight some microfluidics examples from our industrial partnerships. Before I do that, I want to note that virtually all our work is confidential and proprietary. Confidentiality is of prime importance to us. The examples I'll show are all in the public domain, and we do thank our clients for allowing this type of publication. So one of the primary ways that we apply our expertise in advanced multiphysics simulation tools is to test design ideas early in the development cycle, when simulation is often much faster than physical prototyping or testing. Early stage simulation lets us assess feasibility and performance, identify sensitivities and potential failure modes, and evaluate interactions with other system components. And all of this contributes to a better first prototype and meaningful time savings. We've also worked with groups using multiphysics simulation to strengthen concept credibility for IP disclosures, and to support new technology assessments for integration with existing systems. We've worked with groups doing new technology search as well. An example of our simulation supported innovation in medtech and healthcare is our work with 3 Biolabs, and this is a startup that spun out of Mass General Hospital here in Boston. Their scientific founder, Dr. Jay Vacanti, came to us with anatomical drawings and a long-term plan and asked us to provide multiphysics simulation-based design of the internal device channel network, an example of which is shown here. Some of this work was published in 2024 in PNAS, and a patent was granted last summer. Note that our clients own all IP generated as part of our projects together, in addition to all the work product, including models, test data, results, and design recommendations. So this is a rough trajectory of our work together. Early designs tested the 3D biomaterial printing capabilities and also allowed early surgical procedures to be worked out in small animal models. We characterized my colleagues, I'm a fluid mechanics person, but my colleagues characterized the 3D printed biomaterials, and they simulated device loads to provide guidance for robust device design. We use CFD simulations to design the inner channel networks to distribute blood or culture media throughout the device to nourish the cells within. We developed algorithms to automatically generate the CAD for the complex fractal channels, which we then had 3D printed to be tested in vitro and in vivo. So shown towards the right is the more complex device design involving distributor and collector networks with multiple generations of bifurcations. Again, we're trying to mimic the complex nature and performance of the liver. And again, our client work is confidential. We thank 3 Biolabs for allowing us to present this development trajectory. Simulation can help optimize the flow distribution across channel networks. So there was a channel network in the previous example. And this is another one. And in this example, what we're trying to do is evenly distribute flow across an area. One could also not do it evenly, but in some preferential way. But in this case, we wanted to do it evenly. And shown here is a past collaboration with Jonathan Grinham and colleagues at the Weiss Institute at Harvard. We used a COMSOL with MATLAB simulation to iteratively optimize the flow distribution across a microfluidic channel array for a thin wearable cooling system. So here's an example we did with Kevin Healy's group at UC Berkeley. Oxygen transport is a critical design parameter in many biomedical devices that incorporate living cells, and this would include microphysiological systems, lab-on-a-chip platforms, and bioreactors. In the system shown here, and this again was developed by Professor Healy's group, oxygen enters through both gas-permeable materials and inflow. The device consists of a PDMS slab with a media channel, cell culture chamber, and a porous membrane that separates the two. Oxygen is supplied by saturated culture media flowing in to the device, and also by diffusion through the PDMS from ambient air. The simulation allowed us to model the flow, the oxygen transport, and the uptake by liver cells that are cultured in the device, and double check that the flows and shear stresses in the cell culture chamber were tiny. And a key simulation finding was that sufficient oxygen diffuses in from ambient through the PDMS to keep the oxygen concentrations in the media channel and cell chamber nearly at saturated throughout operation. So that means that if a user wanted to use a device to test the zonation, which means high level, the oxygen concentration varies across the device, and you can then see the effect on cells. So if you wanted zonation, if you wanted lower oxygen concentrations, then you'd really have to seal the device from the ambient. So we'll now take a step back and consider the different sub-areas of microfluidic phenomena relevant to industrial problems and how multiphysics modeling can help. So before we jump into how we model microfluidic phenomena, here's a high-level view of the broad area of relevant fluid flow transport and multiphysics problems in which the fluids team here at Verist works. So we work on problems across industry involving flows from microfluidic to turbulent flows, and these are often coupled with heat, chemical or species transport and reactions, things like electrochemistry, solid mechanics, and/or materials science. So we meet the challenge of this broad spectrum of physics with a team of experts, and each person here has a specialized area within these, and we work together with our client to address the problem they're interested in. So a wide range of microfluidic flow phenomena occur in lab scale and portable devices. And so some of these are listed here. Modeling and multiphysics simulations can help understand such phenomena and improve device performance. So showing a few examples, starting from top right, microfluidic devices often or can have successive segments of different solutions flowing through the channel. And this results in reagent dispersion and concentration gradients between solution segments. So dispersion and gradient length increase with flow rate. They actually also depend on geometry, although that's wasn't explored in this particular example. Simulations allow us to estimate the degree of dispersion and the gradient generation and tune the operating parameters to either mitigate or enhance such processes. The example on the lower right is a micropump, and we'll discuss that a little more shortly. On the lower left is a simulation of chemical carryover in the corner of a microchannel. An important step for diagnostic devices involves wash cycles between runs to avoid carrying over chemical species from one run to the next. In some cases, successive reagents are pumped through the device, which are caustic to each other. So in those cases, different samples are run through the device and wash cycles are used to avoid contamination. Simulation can help optimize the wash cycle between solutions. In straight channels, chemical removal improves by increasing the wash buffer volume and flow time. It turns out in corners, there are recirculation zones that can develop, and these increase in size with flow rate. And simulations allow us to optimize such wash cycle strategy based on the channel geometry. So I'll explore this particular area called mixing in more detail in a focus section, but I just wanted to mention it because we're talking about different sub-areas. So mixing in microfluidics can be done by a range of passive and active methods. A few examples are shown below or on the slide. Since microfluidic flows are generally very laminar, efficient mixing often involves chaotic mixing achieved via channel geometry or active methods. Again, we'll talk more about this in a moment. So microfluidic systems often incorporate flexible elements for things like flow control and mixing. Fluid structure interaction simulations can support the design of such flexible elements and the process control around that. The flow around these flexible elements, such as cilia, may be modeled with fluid structure interaction interface, such as the one in COMSOL. In the example here, passive tilted flexible rods act as rectifiers in a micropump, and they're tilted so that there's in the center, you kind of see this peak. This is actually just a piston that's going up and down. And so because they're tilted, as the piston goes up and down, these alternate bending forward and back and the asymmetry they cause means that flow is actually over a cycle, there's net flow going to the right. We actually have a 2D version of this that's included in ComSol's application gallery. Many valveless pump designs are ineffective when the system has low Reynolds number, and so these would be unsuitable for viscous fluids and applications with small length scales. The flexible flaps here uses a dynamic asymmetry to produce net flow. So this works even at low Reynolds numbers, and this has been validated by simulation. So if we go to the top right, a common failure mode in microfluidic devices is delamination between the adjacent device layers. So my colleague Mark Oliver modeled the influence of control channel geometry on the actuation and delamination pressures of this pneumatic microfluidic valve. He used a cohesive zone model to describe the adhesion of the valve membrane to the control layer. And then at the bottom right, this is a lung-on-a-chip microdevice model inspired by the landmark 2010 paper that came out of the Wyss Institute, which mimics the essential features of human and animal lungs. So a flexible membrane, as you can see in the center, separates channels for air and blood, and auxiliary vacuum channels at the on the sides. This enables the membrane stretching to simulate breathing. Modeling the multiphysics behavior of such a device can really complement the development and expedite it. This is a ComSol multiphysics model that incorporates fluid structure interaction, particle tracing, and a nonlinear material model because this was built, or at least originally, in PDMS. And such a model can complement experimental testing and development, and the goal is to reduce lab time and expense. It also allows new ideas to be rapidly tested. So modeling can help elucidate phenomena, also kind of like heat and chemical transport across the membrane, and also cell uptake of nutrients and kind of waste removal as well. So a wide range of industrial processes involve fluid flows coupled to particulate processes such as precipitation, aggregation, and crystallization. The example shown here is a model of lipid nanoparticle precipitation in a serpentine microfluidic mixer for lab scale mRNA vaccine production. So to perform this simulation, we coupled microfluidic flow and transport with the population balance equation. And if you're interested in this type of microfluidic process modeling, my colleague Joe Barakat has a whole webinar on this topic, and you can find that on YouTube. He's also got a live version coming up on August 27th, and sign up is available on our website. Microfluidic devices may involve the interaction and transport of different fluid phases, such as air, blood, mucus, and droplets. For example, the deposition of droplets in the lung airways. And fluid mechanics modeling and multiphysics simulations allow us to analyze a wide range of such phenomena, including multi-phase flows, such as bubbles, droplets, air, liquid segments, cavitation and bubble migration. Bubbles are a big problem, often in fluidic devices. Leakage and seepage, another big problem. Contact angle hysteresis. For those that work in the area of droplets and surface tension and wetting, you may have heard of this, but there isn't just one contact angle. If you take a drop and put it on a surface 10 times, you'll measure different contact angles, and that difference arises from atomic and or molecular and nanoscale and sometimes even microscale pinning and depinning events due to geometric and chemical inhomogeneities. And also, these types of phenomena are certainly present in electrowetting and electrokinetic flows, which are used in a lot of microfluidic devices. So being able to model these is really important to help with the design of devices that incorporate these types of methods. and also to save time. So another important class of microfluidic problems involves of fluid flow to electrochemistry. This combination is central to a growing range of energy and industrial systems, such as flow batteries, fuel cells, and electrolyzers. In all of these, you can understand the performance just by looking at the fluid mechanics or the electrochemistry in isolation. The two are deeply coupled and they need to be together. The example shown here is a proton exchange membrane or PEM electrolyzer, and this is a device that uses electricity, water into hydrogen and oxygen. The exploded view on the left shows the layered architecture with many different components, and they're all sandwiched together. Water enters at the inlet, and the electrochemical reaction is present at the anode, and that's shown in purple. So this splits the water molecules to produce oxygen gas, protons, and electrons. So what makes this a rich multi-physics problem is everything that's happening simultaneously. Liquid water is flowing through the serpentine channel, bipolar plate, oxygen gas is being generated, and it needs to escape through the diffusion layer. Electrochemical reactions occurring at the catalyst surface and current flowing through the solid components. So these are all interacting with each other. The simulation resolves the current density distribution across the active area, the two-phase flow of liquid water and oxygen gas, and also how gas accumulation affects local performance. So this type of model helps engineers understand where performance losses are occurring, how to optimize their channel geometry and the operating conditions, and also how to avoid problems like gas blinding of the catalyst layer. And this is all having to build and test dozens of hardware iterations, although there is a lot of empiricism here, so model validation is always really important. So far, we've discussed fluids like water and other liquids that behave in a relatively straightforward way. If you double the applied stress, you double the flow rate. And these are called Newtonian fluids. Many fluids of practical importance don't behave that way, and modeling them correctly requires capturing their more complex rheology. So non-Newtonian fluids can exhibit a range of behaviors. One example is shear thinning, and this is where the fluid becomes less viscous as it flows faster. Ketchup is a classic example. Shear thickening, where the opposite occurs. Viscoelasticity, where the fluid has both liquid and solid-like characteristics. It can store and release elastic energy. Yield stress behavior, where the fluid won't flow at all until a threshold stress is exceeded. Thixotropy, where the viscosity evolves over time and it depends on flow history. So these behaviors matter enormously for polymer solutions and melts, suspensions and emulsions, foams and liquid crystals, all of which appear regularly in biomedical and manufacturing applications. Two examples on this slide illustrate a range of approaches we use. At the top right, we have a discrete element method model of red blood cells and platelets flowing through microcirculation. Rather than treating blood as a continuum, this approach tracks individual cells explicitly, capturing the rich collective behavior that gives blood its non-Newtonian character at small scales. The bottom example is quite different and practically important for drug delivery. This shows jamming of a dense particle suspension flowing through a syringe needle. As particle volume fraction increases, the suspension viscosity rises dramatically. And at high enough concentrations, the particles transition from lubricated contacts to frictional contacts, which can cause the suspension to jam entirely. The simulation captures where in the needle this transition occurs and how it depends on formulation, directly informing injectable drug product design. Now, a lot of the parameters need to be found experimentally, once that happens, they can then be put into a simulation to test effects of flow rate and geometry. So we'll now do a deeper dive on modeling microfluidic mixing. So before diving into the simulation results, it's worth framing the fluid mechanics that govern mixing at different scales. In laminar flow, the regime typical of benchtop microfluidic devices and systems, mixing occurs purely by molecular diffusion. Diffusion is slow. The characteristic diffusion time scales as length squared over the molecular diffusivity, and this means it grows rapidly. The diffusion time grows rapidly with channel size. So for most species of interest in biomedical applications, diffusion alone is far too slow to be practical. And this is precisely why microfluidic devices rely on chaotic mixing designs to accelerate the process. On the production scale, larger scales, flow is turbulent. So this is not the focus of this webinar, but it's important to consider just as counterpoint. So the physics are different. Turbulent mixing is driven by the cascade and breakdown of large eddies into progressively smaller ones. We can distinguish three mixing regimes: macro and meso mixing, where the timescale depends on the energy dissipation rate; momentum-driven micro mixing with a Kolmogorov timescale; and molecular micro mixing with a Batchelor timescale. So understanding which regime you're operating in drives every downstream modeling and design decision. I think we've had, just see if there's any chat. So there seems to be a question. Thank you for the questions. You can either put them in the chat or in the Q&A. Bubble motion work. Oh, so someone, question about bubble motion work. I'm specifically interested, how do you resolve the air-liquid interfaces in bubble simulations? Do you treat interfaces as a sharp boundary or as a finite diffuse? So that's a great question. And this is a modeling question. We do both, is the short answer. And sometimes we have to decide whether we want to do a back-of-the-envelope model or relying on contact line measurements and just rough estimates, which can be very good because contact angles can vary on a single surface. It can vary in time. You put a surface down, it's all clean, you leave it in the air, it will foul, the contact angle can change. So there's already a lot of variation built in for a single surface. But once you understand that, do some back-of-the-envelope calculations about the force of retention, the force it takes to move a contact line, and that could either be one bounding a bubble on a surface or a droplet on a surface surrounded by air. We can do simulations. There are a number of ways to do that. You can either resolve the surface exactly. So COMSOL has a way of doing that. That would be moving mesh, where the free surface separating the phases, example water and air, is modeled exactly. There's also phase field, where the interface is kind of diffuse. So you use a mesh, and there could be multiple mesh elements across that interface, and there's actually an interface variable. So there are pros and cons of using both methods, and it's really, you want to select the method that is most suitable to the problem, and that's kind of the high level. So we actually have someone, one of our engineers is an expert in multi-phase flows. You'll find some examples on our website. Vial filling is one of them. Droplet, a nozzle with droplets. And so we use ANSYS, Fluent, and COMSOL to do these types of simulations, depending on exactly what's needed. And again, great question, and happy to talk more about that. In fact, the engineer of ours who does those simulations joined us last fall, and we're encouraging him to build his own webinar, so stay tuned. Okay, so I'll keep going, but I'll try to keep checking the chat if there's questions. So here's some equations. These are some of the equations that are under the hood. Our microfluidic simulations, and these aren't all of them. Electrochemistry can add other equations or different types of physics. Solid mechanics would add different types of physics. These are just the ones for fluid flow and transport. So the first set of equations are the Navier-Stokes equations, very old. And these govern the fluid flow. And in plain terms, they express Newton's second law for a fluid. And that balances inertial forces against pressure and viscous stresses. The incompressibility condition on the right, that's this little one, simply says the fluid isn't created or destroyed as it flows through a device. Depending on whether the flow is laminar or turbulent, different terms in this equation dominate. And this is kind of the distinction that we covered on the previous slide. The second set of equations are for mass transport. These are called different things, but one name is the Maxwell-Stefan equations, and this governs how species are transported through the device. These track how the concentration of each species evolves in space and time, and it's driven by both the fluid bulk motion and molecular diffusion. So the key material property here is the mixture diffusivity, which we incorporate as a function of local composition, or we can incorporate as a function of local composition. And critically, these two sets of equations are coupled. Fluid flow drives species transport, and in mixtures where viscosity depends on composition, such as in ethanol and water, which is really important for the mRNA vaccine production and lipid nanoparticle self-assembly systems, the species distribution feeds back into the flow. So this two-way coupling is what makes this a true multiphysics problem. So in microfluidics, fast, efficient mixing is essential in a wide range of applications, from DNA sequencing and point-of-care diagnostics to small batch mRNA vaccine production. The fundamental challenge is that microfluidic flows operate at low Reynolds numbers, where turbulence is absent, and so mixing by diffusion alone is far too slow. So one effective solution is chaotic mixing, where geometric features such as bends, turns, and grooves create complex flow patterns that dramatically accelerate mixing. And this is going to be kind of the focus of this section. So we use multiphysics simulation to simulate and compare different designs, different devices. And in this section, we'll look at three common chaotic mixer designs. So the serpentine mixer routes flow through a series of alternating curves. And in this section, we're also looking at ethanol water. And so you have to make sure that the viscosity, you get the right mixture model for that. We actually have a paper on this. And there are papers that treat this well. One thing to note is that a 50-50 by mass mixture of ethanol water is actually more viscous than either of the pure liquids. So this is something that, if you know about it, it's not a surprise, but it's not necessarily intuitive. So it's important to incorporate that into the simulation. And so in this case, red is pure ethanol, water is blue. And you can see they come in, and there's initially a very sharp interface, and I've shown some cross sections here. And then at high flow rates for the microfluidics, so the Reynolds number, which is the ratio of inertial to viscous forces, is much larger than 1. This inertia drives recirculation in each bend, folding the fluid and progressively homogenizing the mixture along the channel length. So this is fairly straightforward geometry to fabricate. Its performance drops off significantly for mixing at lower flow rates. Simulation, though, you have to be very, sorry about that, very careful, because initially this interface is very sharp. And what you don't want is that your simulation, just by virtue of how it's set up, is adding numerical mixing. You want the mixing in the simulation to be physically based. So we take great pains to make sure that our numerical settings are not the things causing the mixing. So things like mesh resolution, if you know what that is, that we really, we're paranoid that in this case, the settings are going to tell us if we're getting more mixing than actually we would see in the lab. So a second type of chaotic mixer is the ring mixer. This achieves mixing through Dean flows. These are secondary vortices that develop, and you kind of see those in a cross section shown here. As the fluid is pushed outward by centripetal acceleration through each curved loop. And these vortices rotate fluid elements across the channel width. These promote cross-stream mixing, and like the serpentine, it performs best at higher Reynolds numbers. So the third mixer that we'll consider is the herringbone mixer, and this takes a different approach. Asymmetric grooves, and you can see those lining the bottom, and a zoom here. Pattern the channel floor, and this causes the flow to stretch and fold exponentially with distance downstream, regardless of flow rate. This geometry-driven mechanism makes it effective across a much broader range of operating conditions, and this is an important practical advantage when the flow rates either need to vary or need to be kept low. And this slide just shows kind of a zoom of the streamlines and how they move with respect to the grooves. The grooves act a bit like rifling, but then these grooves are alternating also in their orientation, and this helps effectively mix the constituents. So the mixing efficiency can be quantified by mixing time, and this specifically means the time required for the standard deviation of ethanol mass fraction across a channel cross-section to be reduced by half in these examples. So if we look at mixing time as a function of Reynolds number, a clear picture emerges, and we're comparing the three different mixers. The serpentine and ring mixers both achieve short mixing times, but only at higher flow rates where the inertia is strong, and that's just how they operate. At low flow rates, their performance degrades substantially. Results for the herringbone mixer are different. Because its mixing mechanism is geometric rather than inertia driven, it maintains short mixing times across nearly 2 orders of magnitude in Reynolds number. So for applications requiring mixing at low or variable flow rates, this is a significant advantage. So the broader takeaway actually is that simulation gives us the ability to compare mixer architectures and optimize geometry and operating conditions before committing to fabrication. And this reduces the development time and cost. It can also help us figure out why we're getting better mixing, because you can actually look at the streamlines in the simulation. Let's see, there's some more questions. Have you modeled drug delivery systems that mix fluid with lyophilized beads, looking at how different designs may impact bioavailability? So yes, in short, we have. In fact, we have a February webinar that's on YouTube all about combination products. One of the examples we looked at there, and actually Joe's particle transport webinar that I mentioned earlier, you can either find that on our website, but it's also on YouTube. Those one of the examples that Joe's worked through, what happened are dissolution of beads. Now they could be lyophilized beads, they could be just other types of reagent beads. We certainly do a fair amount of work at different scales, both in production scales, but also in small scales, looking at these particles dissolving. They could be porous, they may not be porous, and then the solute goes from the undissolved state to dissolved, and then we have to track that within the dissolved chemical or species within the fluid and how that moves around. So we can certainly do that both in either COMSOL or ANSYS, depending on if there's turbulence, depending on how intense, if there's turbulent intensity. But we certainly have examples on our website, also a case study example of the dissolution. It's actually, I think it's called reconstitution of a drug product. Would it be possible to design and optimize a structure if you'd like to use phase transition for cooling? Sure. We certainly do phase change for both cooling and other applications. And we often compare geometry. I mean, this geometry is an input to models. So we can, once that's in model form, we can parameterize those geometries and we can do sweeps over the geometry to figure out, which one's going to give the best performance. And we can also understand why by looking at streamlines and temperature along the flow streamlines, or where areas may not be getting enough flow. So please reach out to us if that's an interest. How do you define a homogeneous mixture in simulation? Is there a threshold? Right. So there's different ways to assess homogeneity. One is just the coefficient of variation of either the mass fraction or the concentration. That's kind of what's plotted on this slide. If you were looking at temperature, then if there's an important parameter you want to track, and you want to make sure the mixing is finished to a certain degree, you'd want to track the variation of that in a cross section. You can easily do this with simulation. Okay. Okay, there's a question about boundary conditions at an inlet. How can you calculate Reynolds number, different velocity? Oh, yes, sure. A great question. So we can certainly calculate the Reynolds. So the question is, how do you calculate Reynolds number when you have two inlets? There are different flow rates, different viscosities. So the answer is, so there is a different Reynolds. You can certainly calculate that the Reynolds number at the inlet, and then at each inlet it's going to be involve the flow, the average, either the flow rate, the cross section of that inlet, the viscosity of that particular solution. Downstream, though, at every point in the fluid, we have a mixture model for the viscosity. So we know what the viscosity is at every point in space in the device. And that's important because the model wouldn't run if we didn't have that. So with that viscosity, you can take a cross section and you can say, okay, I know what the speed is at every point, because that's what the simulation is calculating. I know what the viscosity is at every point in space. And then I can say, okay, I'm going to average over a certain cross section. And so that's how we can get the Reynolds number. I think this case compared to the same value. I mean, CV, you know what the concentration and mass fractions are at every point in space also. So you can just in COMSOL, it's very easy to say, okay, at this cross section, I'm just going to calculate the standard deviation and mean, because they're just integrals. They involve integrals across sections. So that's straightforward to do. Happy to talk about that further. Okay, so I had an example. We have about 13 minutes left. This is kind of the last example that I was going to work through. And this example it's a nice example. It's an old one. I did this with my colleagues at Harvard Medical School when I was a postdoc there. But the nice thing is people often think a simulation is just, okay, we open our simulation software, COMSOL or what have you, we load in a geometry, we set the parameters, we choose the physics, and we run, make sure that all the settings are chosen, we do a mesh resolution study, and we get results. That's true, but there's a lot more that you can do, and this is the beauty of modeling. And so in this example, we combine different modeling tools. So this particular example is a grooved microchannel. The application is one of tissue engineering. We were trying to do patterning, and this was a double grooves that my colleagues were testing. There are cardiac cells here, and the flow, because there's circulating flow, you can actually line up. Particles will get deposited in the corners, and it turns out with these cardiomyocyte cells, when they are beside each other, they'll actually join and form fibers that can then start beating. So it's a nice a classic tissue engineering example. My colleagues were wondering, well, can we adjust the flow so that, and they'd found experimentally that they could adjust the flow and they could get upstream or downstream cell docking, they called it. So depending on the flow rate, depending on the groove depth and aspect ratio, they could get the cells to line up in different grooves. Well, this is a beautiful fluid mechanics problem, and you can actually solve it with simulation plus a little bit of extra fluid mechanics tools. And so we used a range of tools here. We used to certainly understand the physical picture. We also used dimensional analysis, which is taught to every fluid mechanics student. We used simulations to make a phase diagram, and then we did some validation. So the physical picture is that in shallow grooves, the flow enters and exits without significant variation. It just goes in and out without significant recirculation. Cells dock, in this case, in the downstream corners. In deep grooves, you can get recirculation, and in this case, cells dock near the upstream corner. So, so that's the physical picture. Now we want to do dimensional analysis, and the reason this is important is that there's 7 for a single groove, and this is a simpler case than the double groove. There's actually 7 physical parameters. 4 lengths, a speed, a density, and a viscosity. Now, it turns out that we can recast this in a dimensionless system, and there are only 4 dimensionless parameters. There's a theorem, it's Buckingham Pi's theorem. And it turns out there is no approximation. This is exact. And it's just that nature doesn't care whether you're measuring length in feet or meters or centimeters. It is just a length, and it doesn't care whether you measure. There's nothing special about meters per second, feet per hour. These are just quantities. And so it turns out that that principle allows us to recast the system in a dimensionless form. And we can actually be clever about how we pick the dimensionless numbers. One's a Reynolds number because we know that has a significant physical meaning. And the other are geometric, 3 aspect ratios in the problem. So that's just dimensional analysis. So immediately, without approximation, we've gone from 7 parameters down to 4. And so if you had to go in and vary these parameters experimentally or in a simulation, you don't have to vary 7 independent parameters. You can actually just vary 4. So then we built a COMSOL simulation. This is fairly straightforward. Just in this case, it's just 2D fluid flow. This is technically not multiphysics, single physics here. And what we looked at, we asked the question, when do you get recirculation? And there's an easy way to test for that. You just can look at the sign of the shear stress on the bottom. We said, when is the flow going in and out, and when is the recirculation? And then we can vary the aspect ratios, 2 of the aspect ratios, and then we can plot a line, what we call a phase boundary, when that behavior changes. Then we can redo these simulations for different Reynolds numbers. Now, the one thing we didn't vary was the length. We just assumed this L naught was long enough. So we're not worried about this parameter here. We're just varying the others. And lo and behold, we find this phase boundary, and the experiments collapse on either side. So now we have a high-level description of what happens, of how this system operates. And this is a great insight. There's only two parameters that are important. In microfluidics, Reynolds number is generally small, 1 or less. And for these different Reynolds numbers, all these phase boundaries coincide. So Reynolds number, for the purpose of understanding the behavior of this system, Reynolds number is not important. That means you don't have to worry about it. That means there's only two aspect ratios: the channel height over the groove width, and the groove height over the groove width. Those are important. So we can use modeling to simplify our understanding of the system, and that is huge when it comes to product development. We did the same thing for double groove. High level is just that out of 9 physical parameters, only 4 that are actually important, and these are all dimensionless, of course. So I see lots more questions. I'm going to answer these. I want to thank you for your attention. We'll now do some Q&A. I would kindly ask you a favor. Please fill out our 3-question questionnaires really quick, either through the QR code or this link. If you'd like to see additional content in the future, please let us know. So now let's go through some questions. Okay. Someone's working on their master's thesis on a diamond microfluidic cooling device, CFD simulations to optimize mixing between the cold fluid core and the warmer fluid near the walls. Channel's very narrow, and what type of mixing structures or geometries? So that's a great question. If in this case, if you do, so in the simulations we did that I mentioned earlier, we were actually just trying to get an even flow. But if you have, say, maybe the sides of the device are hot, maybe you actually want to direct more flow towards those. So when it comes to selecting, which structures or geometries, that's probably start with the ones that you can reliably fabricate. Because one thing, certainly, if I don't know if this is a flexible device or not, but if it is, in the case we were looking at, it was. And so actually you could get bowing of the channel cross section. The problem there is that then that really changes your fluid flow, because the flow resistance of a wide rectangular channel scales as 1 over the height of that, the thin height cubed. So if you double the height of your microfluidic channel, the wide microfluidic channel, the flow resistance goes down by 8-fold, almost an order of magnitude. For a square channel, it's even worse. It actually goes down 16-fold if you increase its size. So if you're getting, if your channel cross sections are changing, if it's made out of a flexible material, you're going to want to include that in your model. And maybe initially, the type of model maybe isn't in 3D. Maybe it's kind of like a flow network model. There's an analogy between electric circuits and flow networks, and those help kind of narrow down the range of kind of the channel network layout. This brings up an interesting challenge, because in a lot of our work, it's not just what the simulation says. We can make lots of recommendations, but those recommendations have to be tuned to what the challenges that our clients are facing, and manufacturability is certainly one of them. So that is I think the answer to your question is much longer than the time that we have. But if you have a simulation, you can test different designs. But you want to ask things like, how easy is it to make in the lab? Is this out of a rigid material, or is it a flexible material? If it's flexible, how do I know that the channels are actually what the CAD is and not something else? So that's actually what I would look for And certainly one of the challenges my colleagues faced was delamination, because, you know, the simulation can change the shape, but you have to make sure there's enough, where there's bonding, you have to make sure that those bonding regions are big enough that you're not going to get delamination. So it's a great question. Modeling can help, but certainly in the context, you want to do it in the context of all the other criteria that you have to work with. Let's see, is there any Q&A? So I think that is pretty much it. We're almost at the top of the hour. Certainly, if you have questions later, you can even add questions on the survey, or you can just shoot us an email at contact@Verist, or you can certainly find my email is mhancock@Verist. So feel free to shoot me an email if you have questions. This webinar will be available. Has been recorded. It's available for viewing later. So if you have things that you'd like to look at, or can certainly do that. So thank you, everyone. Appreciate everyone's attendance and all the questions. Those are great. Have a great rest of your day.